Thanks a lot, and thanks a lot to Dappa and Sumantra for organizing all this. I'm really sorry I can't be there. I wish I was, and I hope I get the time or the chance to visit ICTS at another time.
I just want to interrupt
yeah.
Christina. Christina is also a co-organizer, so it's not just me and Sumantra. So Christina did a lot of work, even if she is not here by person. So I just not just us.
No worries. Okay. All right, then. So let me start with just giving you a little overview of what I've been working on over the past years. So very generally, I'm interested in theory, I use tools from statistical physics and fluid mechanics to address different topics in soft matter and more specifically active matter systems. So you've heard a lot already, I think, in Thomas' lecture about how we characterize the dynamics of single active agents like Janus colloids. And today I'll focus a bit more on E. coli bacteria. And more recently also got involved in projects that include crowding and transporting porous media. In addition to that, I'm sort of interested in elastic properties of polymers. And it's quite interesting because theoretically, the concepts of describing a semi-flexible polymer is very much related to trajectories of active particles. And I'm not sure if Thomas mentioned this, but that's an interesting theoretical fact. And then finally, I'm working on fluid structure interactions that involve hydrodynamic coupling between particles and, for example, surfaces that have a particular structure or are deformable, as you may have seen on Sagnik's poster the past days. So let me start with just giving you an overview of different types of microorganisms. And I think it's in fact quite beautiful that although all these organisms I'm showing here come from different domains of life, they have evolved very similar strategies to self-propel and move in liquid environments. So here, for example, you have bacteria, an E. coli bacterium with several flagella attached to its back. And these flagella rotate and they form a bundle and they allow the cells to swim. Similar concepts are also employed by archaea. So here's an example where you see the flagella. And in this area, they are often referred to as archaea. So they got a different name because it's a different sort of domain of life. Anyways, and then there is this broad group of eukaryotes that involve protozoa that have these, like paramecium, that have these tiny cilia attached to their surface, and they cooperatively generate traveling waves and so lead to self-propulsion of cells. There are sperm cells that have an individual flagellum, or also these algae, which have two that are attached to the head, and that leads to, they do something like we do when we swim in water. So now not only can these microorganisms swim, in fact, most of them have some way that allows them to change their swimming direction. And that's important, for example, to forage food or to escape from harm. And the most prominent one is certainly the E. coli bacterium. And here you can see a movie, which essentially moves straight over a long time. And at random times, it then just changes the swimming direction randomly. And I'll talk about details of this in a bit. But you should know that there are also many other organisms, microorganisms that employ different types of reorientation mechanisms. And here you can see one where the cells actually perform this run-reverse motion, right? So they move straight, they change orientation most likely because of some rotational diffusion, and then they sort of reverse and move backward in time. And so now here in color, you can see these are species that are from these that belong to bacteria, to eukaryotes, but also to the class of archaea. Recently we actually made a list of different species just with the purpose of trying to, characterize them in terms of body lengths and flagella. But then we noticed that actually almost all of them have some sort of reorientation mechanisms, be it run and tumble or run reverse, but it can be also other modes that are a combination of the two and similar. So just so that this is very prominent in nature. So now let me give you a brief overview of the talk. So I'll show you a quantitative way of how we can characterize run and tumble motion of E. coli bacteria. And then we'll use this tool that we developed to actually try to relate aspects of the bacterial motor that drives the rotation of these flagella to the swimming dynamics of cells at the population scale. So you can imagine in this case, we have to bridge across several scales, and that's kind of I find an interesting research question. And in the second part, I will move towards the idea that microorganisms, in fact, usually don't move in dilute environments, but they interact with their natural habitats. And I'll sort of focus on two aspects. The first one is external cues that can change the swimming direction of the cells, and I'll show you a minimal model on that. And in the second part, we'll talk about run and tumble behavior in porous media, which sort of adds to what I'm sure Tapa has shown you already. So let's get started, and let's look again at this video from Howard Berg's lab. So here you can see these cells, they move, and perhaps if you look carefully, at some point they change the swimming direction because the flagella bundle essentially dissolves. So now we can, so thinking about this a bit more carefully, the way this works is that the cell, as I said, has these multiple flagella that are attached to the body, and they form a bundle, right? And they form a bundle exactly when all the motors that rotate the flagella turn in the counterclockwise way. And then during a tumbling event, what happens is that one or more of these flagella motors start to spin in the opposite direction, in the clockwise direction, and that leads to a tumbling event. And this tumbling event takes a while until, all the motors start spinning in the counterclockwise direction again. And so now this whole process of relies on a sort of complex chemical machinery that is responsible for the rotation of the motors. And I'll just give you like some really basic molecular mechanisms and focus actually on two proteins that I know about, but I'm certainly not an expert on this whole chemotactic signaling pathway that these cells have. So now one way to sort of study how this flagella rotation is related to the motor is in terms of these type of experiments where people have sort of attached cells to a glass plate, and they attach a bead to the flagellum. So now they can do two things simultaneously. First of all, they can label certain molecular species and actually sort of measure the concentration of those inside the cell. And second, by tracking the bead, they can actually know or learn, measure in which direction the motor is spinning. And so in this experiment, they were looking particularly at the concentration of phosphorylated KY. And here it turns out if there's a lot of KYP present in the cell, the bias for the motors to rotate in a clockwise way approaches 1. So that means if there's a lot of KYP present in the cells tend to tumble, right? And that's the one thing I'd like you to remember. The second thing that you can, or the second component in this whole machinery that's important is actually KYC. So now KYC, this phosphorylates KYP to KY, and that essentially means that it suppresses tumbling, right? So if there's less KYP present in the cell, the bias for clockwise rotation of the motor becomes smaller in principle. And so this whole picture of when the transition between a tumbling phase and the run phase occurs is the picture that we have is that if the cells are able to express KC, they should be able to actually transition from a tumbling state to a run phase. And this is sort of the aspect that we'll focus on in this work. But before we can actually look into these strains, we'll need to sort of find a way to quantitatively measure the run and tumble statistics of E. coli in 3 dimensions. And to do that, we sort of collaborated a while ago with a group of people that include Yongfeng Zhao and Julien Tailleur, together with Thomas Franisch on the theory side. And the experiments were done actually in the group of Wilson Poon at the University of Edinburgh, and they were led by Vincent Martinez. So now this work has been going on for a while, but I think we finally made it. And so let me briefly explain to you how we measure these statistics. I'm sure you've heard already by Thomas Fanos about this method of differential dynamic microscopy, but I'll still sort of repeat some aspects of it for you. So this method is sort of a complementary method to particle tracking, and it relies on, rather than actually tracking individual cells, on measuring image intensities and correlations among those. And the way this is done, that images are taken just as if you were doing particle tracking, and then these images are sort of subtracted from one another so that one can measure the density fluctuations in the sample. So this is, and this whole thing is done in Fourier space and correlated. So I don't want to go into details, but there are like three if this image processing is being done, this function G here is related to, first of all, the signal amplitude and the noise in the system. So these are two quantities that can in principle be measured from the experiment. And then it's related to this intermediate scattering function. And I'm sure you've heard of this already. But so the intermediate scattering function is sort of the exponential of minus ik, where k is the wave vector, times the displacement variable. And so what it does, it measures the dynamics at length scales 2 pi over k and at length times tau. And for those of you who work on theory, this is the characteristic function of the random displacement variable. Okay, so what we're going to do next is I'll show you a way to compute this function theoretically, and how we can then compare that to experimental results. Okay, so now in terms of theory, let me just start with a really simple run and tumble model. And in this model, what we assume is essentially that during a run phase, the cells move straight and they randomly change their swimming direction at tumbling events. And this happens at exponentially distributed times. Okay, so now if you look at these dynamics at the larger scale, what you expect is actually a random walk, an effective random walk with an effective diffusivity that depends on the speed of the cells and this tumbling frequency. Okay, and so we can write this process in terms of a renewal theory. And here you can see two equations. One is for P. So P is the probability that the cell displaces a distance r during a lag time tau. And the other one is actually for the probability that a renewal event, in this case, a tumbling event, takes place at the position r and the time tau. Okay, and so this P depends, first of all, on the probability that the cells are at the position r at time tau without ever having tumbled. And so now this is important because if you do your experiments, you can imagine that you don't start measuring at the point where each of the cells is just starting their run, but you start essentially in a stationary state. Okay, so we have to account for the zeros for this probability separately. Okay, and then the second term sort of involves the probability that the cell has tumbled at an earlier space at an earlier time, times the probability that the cell keeps running. So phi here is the runtime distribution, and times the propagator P, which tells us the probability for the cells to displace during their run, right? And to move a distance L during time T. And so now similarly, we need an equation for Q. And Q again depends on the probability to tumble for the first time, Q1, plus the sum over all other events. So here we again have the probability that the cells have tumbled at an earlier space at an earlier time, times the probability that the run ends exactly at this time point, and the probability that the cells have moved during that time. Okay. do let me know if you have any questions, right? Okay, so now we have these two equations, which perhaps look a bit complicated, but there's one thing we can notice immediately. We have a convolution product. So now how do we get rid of this? We can essentially perform a spatial Fourier transform, and we come up with two equations, which look much simpler, but also you will notice that they can be solved for each wave number separately, right? And what you should, what's also important is that in fact, one of the equations for the probability to displace now, it turns into an equation for the intermediate scattering function. And that's the one that we are interested in. Okay. And so now what we're gonna do, we solve these two equations numerically for different wave numbers, and we will then compare this to experiments. So let me add to this. So reality is in fact more complex. A simple run and tumble model wouldn't do the job to really analyze this this biological system. And so one aspect that's always important in experiments is that you will always have a non-motel fraction of cells. And this non-motel fraction of cells has to be included in our theory. So overall, the ISF is then a sum of the ISF for run and tumble cells plus that for passive cells, right? So e to the minus d k squared tau is the intermediate scattering function for cells that are just simply diffusing. That's an assumption that we take and that sort of seems to work quite well. So now there's another aspect which is important, and that is that cells usually take some finite time to tumble, okay? So they don't tumble instantaneously. So what that then means is that we have 4 of the equation that I've shown you before, because we'll have to couple to the tumbling process. And during the tumbling process, we would assume that the cells are diffusing. Okay. And then finally, and that it's been observed in experiments, but single cell tracking, that the E. coli cells actually exhibit a large velocity distribution. And so we have to account for the fact that cells have different, or the speed of cells changes over time. Okay, so now let's look at one of these theoretical ISFs to sort of understand the features of them, right? So here you see the plot of the ISF as a function of time for different wave numbers. And there are a few features I'd like you to notice. So first of all, so the solid line here is for cells that don't, that tumble instantaneously. And we see that at high wave numbers, we have these oscillations. Now these oscillations, if we see them, they are typically a fingerprint that the cells are swimming. Okay, so now if cells actually do need some time to tumble, we start seeing these plateaus. And so if you see a plateau, and there's even some hint of oscillations, if you like, that's a sort of a fingerprint that the cells are tumbling. And then you might see that at long times, there's still a plateau, and that sort of accounts for all the non-motel cells that are present in the sample. Okay. So by just looking at these features, we can already sort of try to understand some aspects that are happening within the sample itself. So to actually compare to experiments, we've validated our theory with simulations. So what we did was we performed agent-based simulations that reflect the experimental setup. And then we developed a fitting protocol to retrieve the motility parameters of those cells, okay? And here you just see a typical plot of such a fit. I should note that it's not so easy actually to fit these data, and it's really important that the data are that experimentally one can measure data that cover a large range of time and length scales. Okay, so we need to recover length scales that go down to where cellular diffusion is important, up to length scales that cover several times the persistence length of the cells, right? Okay, and so let's now move to actual experiments. So here on the right, you can see the part of the symbols that are experiments, and we shifted the ISFs just so that you can actually see the different features. I should say we were able to measure the ISF over length scales that cover a few microns to a couple of 100 micrometers, which is quite a large range. So within the sample, we had about 10 to the 4 to 10 to the 6 cells. And when we then, so that provided us really good statistics. And then fitting the data to our theory, we can actually really see very nice quantitative agreement between them. And what does that give us? So that essentially gives us access to the motility parameters of the cells. So we can now, from this, extract the speed of the cells, which is about 16 microns per second. That's typical for E. coli. The translational diffusivity, which we find as 0.24, which agrees with sort of a prediction if you just consider simple Stokes-Einstein relation. And then Motel fraction, which is actually really high. So now these three parameters have been done before, so that there have been several ways to get that quite rapidly. But what has been done, or has been really hard, was to extract the run and tumbling times. And these ones we were able to measure. So these are, so we have a run time of 2.4 seconds and a tumbling time of 0.4 seconds. So now, as I told you, at large scale, the system is expected to become like a random walk. So we can calculate this effective diffusivity. And here you see that it actually depends on the sum of the velocity squared plus the fluctuations of the velocity itself. And so we come up with about 200 micrometers squared per second. And the speed fluctuations actually take about, I think, 20% of that diffusion coefficient. To sort of do a check on whether what we find here actually is reasonable, we can look at the intermediate scattering function at even larger scales. And what we see at these large scales is that they relax like simple exponentials. And so we can sort of fit this data to an exponential decaying function with an effective diffusivity. And by just looking at that, we actually get an effective diffusivity, which very well agrees with the one that I just showed you. So I think that's fine. So let's move on to, or some of you who know Berg's work might actually ask, how does that compare to what we have found? So now in Berg's work, they looked at 3D tracking actually of about 35 cells. And they find mean tumbling time, which is about 0.14, and the mean run length, which is about 0.86, which are a bit different to what we see, but overall they see a fraction of time running, which is about 0.86. And this is perhaps per coincidence a bit, but it's exactly the same number that we also see. So I think like overall, the order seems, or like the quantities that we get seem sort of fine compared to what has been done before. And even if people nowadays track in 3D, they get values that are quite similar. So anyways, so now that we have a method that we think works, we can move to some engineered strains. So now here, as I've told you before, we're sort of interested in this transition of how cells change from a clockwise rotation of their motor, like from a tumbling state to a run phase. And the question that we ask is if we can engineer E. coli in a way so that we can tune this tumbling behavior, okay? And so what has been done by Nansu in China, so he was able to engineer such a cell. And what he exactly did was that he deleted the key Z gene of the cell, but added an inducible plasmid. And sort of what the plasmid does is the following: if the cells grow in a medium where IPTG is present, the cells this plasmid restores the expression of key C. and the rate depends on the concentration of IPTG. Okay, so at the single cell level, this has been checked, and it seems indeed that the expression of Kc can be tuned with IPTG. So now our hypothesis is, well, if cells are grown in an environment with a low IPTG concentration, they would be tumbling all the time. But if they grow in an environment with a high IPTG concentration, they would be swimming. Okay, and so we can look at these ISFs again for now samples with different IPTG concentration and at different length scales. And you will see that there are actually, so we look at IPTG 0, 25, and 150 micromolar, and you see three distinct, or essentially two distinct features. So the ones with a high IPTG actually have these oscillations, and I told you before that oscillations are sort of a fingerprint for swimming motion of the cells. And at IPTG 0, we see these simple relaxing exponentials. So we would think that the cells are tumbling in that case. So now, qualitatively, I think we already see what we were we were hoping for. But now we can also really do quantitative fits of these experiments. And we indeed find something interesting. So we find, first of all, that as we vary IPTG, the speed of the cell stays the same. Okay, this is important. So irrespective of IPTG, the cells swim at the same speed, more or less. I should say here we did like two experiments, experiments with two different biological replica. Yeah. And then in the second, yeah.
Hi, Christina. So IPTG is essentially a crowding agent, right?
I don't know. What do you mean by crowding agent? Sorry.
So basically, does that increase, so like the passive particle that hinders the motion of the bacteria?
I don't think that it affects the motion of the bacteria.
Okay.
It's dilute enough. And also, no, but I should say, they grow in this environment, and then the cells are actually taken out, and they are put in an environment where they can swim, and then we measure
I
that.
think IPTG is a sort of like an energy source, like a sugar, right? I mean, so people use like engineered bacteria that can metabolize this specific sugar and does other things. So I think what they are doing is basically they programmed a bacteria which can use this as the primary source of carbon and then go from there.
Yeah, but only during the growth phase. And then they're resuspended in a different medium. Does that make sense?
Excuse me, ma'am. Hello?
Yeah, sure.
So in your theory of run and tumble dynamics, so what waiting time distribution have you chosen?
In both cases, exponentially distributed ones. We did check different distributions, but first of all, those mostly come with another fitting parameter. And second, I don't think these ISFs are really sensitive to the distributions at the length scales that we look at and in the parameter range that apply to E. coli. You can tune them in a way. For example, if you have a wide velocity distribution, you can get different features at large scales. But for the parameters of E. coli, I don't think the experiments are sensitive to extract that information.
Okay, but there are some papers where I have seen the power law distribution for this waiting time distribution. So why not that?
So we did that too, and the results are not changing. So the only thing that I can confidently say and predict with this is actually an average runtime and an average tumbling time.
Okay, so waiting time distribution is actually the Poissonian distribution, right?
Also, yeah. Mm-hmm.
Oh, okay, thank you.
No worries. It's a good question, and it's debated a lot, but I don't think we are able to distinguish that. Okay, are there any other questions or?
Is it experimentally observed that they follow exponential distribution?
Well, it's debated. I think, well, as what was said just before, like some claim that there's some power law or like some log normal distribution. I think Berg noticed earlier that it's an exponential, and that's what people have been using mostly. But it's certainly something that's still open, I think, to some extent. Any other questions?
Hello. Yeah.
Yeah.
Can you please also explain how these undulation in the FKT signifies swimming?
Okay. Yeah, sure. So, okay. So if you think just of a particle that goes in a straight line, right? So the displacement is just the velocity times time. Okay. So the distribution of that would be a delta function, right? So you have a delta. I'm sorry, I can't write it now, but it's a delta of your r minus vt. And so because if you do the Fourier transform of that in an isotropic system, you come up with in 3D a function which is the sinc function. So it's the sine of vkt divided by vkt, right? So mathematically, that's where the oscillations come from. Does that make sense?
Thanks.
Yeah, okay. Any other questions or? Okay, so I think I'll move on.
You can continue here.
Yeah. Okay, good. So the last property that we wanted to look at in this respect is actually the persistence length. So now the persistence length is here shown as a function of IPTG again, and it's defined as the product of the velocity of the cells times the runtime, okay? And so we do see as the IPTG is varied an increase of the persistence length, okay? And that goes from small up to 150 or so. So the last step, as I said, we wanted to actually relate that to the Kc concentration inside the cell. And this has been measured independently. So here is the count of the Kc messenger RNA per cell as a function of IPTG. And we do see this increase as well. So now correlating these two, we in fact find that as you increase Kc, or as more Kc is expressed, we actually have a larger persistence length. Okay, so now let me just sum it up. So I showed you a quantitative way to characterize the run and tumble dynamics of E. coli. And we can actually control these run and tumble dynamics now with an engineered strain. Okay, so and this engineered strain really allows us to sort of, for example, study and test active matter theories where you're sort of interested in a system where the speed remains the same, but you just want to know how the tumbling rate actually changes the physical picture. And this hasn't been there so far. So far, people were really able to tune the speed of the cells or both of the things simultaneously. Okay. So now, are there any more questions or are we good? Because then I move to the next topic.
For this engineered bacteria, like you have shown a cell. What about if we have a robot?
So you have to be a bit more specific. Like, what do you mean? Like you mean just
like if like robots can be programmed to perform run and tumble motion, and like they can be also easily tuned with different parameters like diffusion constant. So like
okay, so I don't actually, and perhaps you can send me that, but I'm not aware of like actually micro robots that you can tune, where you can actually tune the tumbling behavior. I know that you can tune the speed and everything.
like our lab is actually like working on this. So we have a paper on archive.
Okay.
So we have like made these programmable robots, which can be programmed to perform different kinds of motion, like Brownian motion, active Brownian, or run and tumble motion. So like if we see their mean square displacement and certain other parameters, they actually show the behavior that is shown by a bacteria, or like which shows run and tumble motion.
Oh, excellent. Cool. So what was your question about that?
I was just saying, like, what will you say about that? Like, any comments?
Well, I would think that there's one thing you can do. If you really want to distinguish whether ABP, an active Brownian particle and a run and tumble particle or if you want to distinguish the two, let's say, right? We know that the MSD is exactly the same. So that's insensitive to either you tumble or you do rotational diffusion. But what you could look at is actually these scattering functions, because the scattering functions, they are sensitive to this difference. We have a paper on this, and I think Thomas talked about this, the ISF for an ABP. And if you look at that paper, we actually try to, or we show that for ABPs, like simultaneous colloids, their ISF really corresponds to that of an ABP and not to that of a run and tumble particle. So essentially, you could use these theories to really distinguish, if you cannot see it by eye, for example, or distinguish quantitatively what actually motion those particles are doing. Does that make sense?
Okay.
So I guess what I'm trying to say is that the MSD is insensitive to these details of how particles actually move. But if you move towards these intermediate scattering functions, you can distinguish these aspects. And what I should also say, like if you're in 2D and you can easily track, you don't have to do this DDM, but you can just track the cells and still compute the same quantity from the tracks. Yeah.
Okay, thank you.
You're welcome.
Hello.
Yeah, hi.
So this intermediate scattering function, can it be used in a 3D system for?
Either. I mean, the one that we have here, the cells move in 3D. So we have a theory for 3D random tumble particles, but you can also do it in 2D. If you say have colloids moving at the surface or bacteria moving at the surface.
Hi, Christina, this is Tapa. So, so when you say like you have the DDM for 3D, so if you're imaging this, is it like a volumetric image that you do? And but I thought like it's always like a 2D projection, and you change the scale. So the 3-dimensional information is embedded in the projection in 2 dimension.
Yes.
Okay.
Yes, you're absolutely right. So we just take a large depth of view, which is that of several persistence length of the bacteria. And that's always debated. This is a very good question. But what we did to sort of rationalize that we can do this actually to like computer simulations, trying to make images from the simulations as you would do in experiments and extract parameters, and it worked. Yeah.
So if I have in a not so fast moving system, for example, not bacteria, maybe some sort of mammalian cell, and I can take like volumes of image, like Z stacks or something like that, and I have multiple times, I can in principle also do DDM but I'm not sure how to think about the intermediate scattering function in that context.
Okay, I get your point. I think we would need to properly derive what this differential image correlation function, so like the output of DDM would actually give you. Okay. So, and then how that is then related to the ISF. Yeah, I think
okay. Awesome. Thank you.
I can tell you right now. Okay.
Any other questions? Yeah, let Christina finish, and then we can take another set of questions.
Okay, perfect. So, because I thought, so what I briefly gonna show you is how we can use this model that I showed you to actually think about external cues and how to model, for example, a chemotactic bacterium. But before going into detail, let me just introduce to you a bunch of environments where microorganisms are present. And so, as Tapomoy have already told you, like many microorganisms live in soil. And in soil, they move in really tight spaces, and so they interact with their boundaries. And so it's interesting to study how these cells move in porous materials, for example. Many microorganisms are present in the ocean. In the ocean, they are subject to large flows and chemical gradients and fields. So that's another topic of research. And finally, there are many microorganisms in our body, and here you can see an image of a root canal, so of our tooth, and where cells actually penetrate, and other cells can also sort of like squeeze through tissues. So now in all these environments, cells are really subject to different environmental complexities that include flows, confinement, chemical gradients, complex fluids, and so on. And what I'm going to show you now is sort of a model how we can think about particles moving in the presence of external stimuli that can change the orientation of the cells. So now the most prominent example is a chemotactic one, where you have cells that adapt their swimming direction and the tumbling frequency in the presence of a chemical species. And so that allows them to move towards food gradients. So another example that I found along that line is that actually biofilms that are composed of bacteria can attract distant cells via generating electric signals. And so these signals tend to change the tumbling behavior of the cells so that they eventually come closer to the biofilm. And that actually, this mechanism also works across different species, if you like. And so now a different way to tune orientation, but we already heard that this can be done for synthetic particles. You can also have them run and tumble. But here there is another example where Janus colloids were subject to different magnetic fields, and these magnetic fields sort of change the rotational diffusivity of the Janus particles. So now, since inactive matter or inactive particles, the change of orientation is always coupled to its position or the change in position. So there are a lot of interesting phenomena that can be observed. And so what we were interested in was just a simple model where we think about an active particle whose swimming direction is aligned with some sort of external stimuli at random times. And so the idea is that in between those two, let's say, resetting events where the particle reorients, the particle actually performs active Brownian dynamics. And you've heard of this a lot already, but in a simple ABP model, the particle moves along its orientation at a fixed speed, and the orientation is subject to rotational diffusion, right? And the position as well to translational diffusion. And so now in our model, we reset the swimming direction of the particle at random times that go with the distribution phi of t, and we reset it to a resetting angle theta r. And there's sort of an average resetting rate lambda. And so overall, we have a reduced resetting rate that is sort of the average resetting rate with respect to rotational diffusion. And we'll look at some aspects along this line. So now the way we sort of model this is in terms of the same equations that I've shown you before. I just now write out the propagator for the displacement explicitly, okay? So again, we have the probability to displace. The distance r during time t, that depends on this probability to never have tumbled. And then it depends on this runtime, or in our case now resetting distribution. And here we have the probability of an ABP now to move a certain distance L during some time t, having started with an initial orientation and a theta naught, okay? And here P of theta naught and theta r is the distribution of the angle at the beginning of the resetting event. And so now we assume that at each resetting event, the particle starts out with the same angle. So we'll just have a delta distribution. So now overall, this long term here, where we sort of then integrate over the angles, gives us the probability to move a distance L during time t, given that after a resetting event, we have a specific resetting direction. And so now one can look at the same equation for Q. So now the probability for the last resetting event, again, depends on this Q1, and then sort of on the sum over all previous resetting events, like what we've shown earlier. So now, if you recall last week, Thomas showed you that we can actually compute the propagator, right, for the active Brownian particle model. And he showed you that this can be done analytically, and you can do that in terms of these Mathieu functions. And this will be now input to that theory that I'm going to show you. Okay. So now these equations, as we've done before, we can move to Fourier space. And in Fourier space, they now depend on the wave vector, or like they depend now on the wave vector k. And we have like this equation that gives us again an intermediate scattering function. And the interest, or like the part which I have to emphasize is here, that is the P, like that one we know what we can tune or change the resetting distribution as you asked before. So that can be an exponential, that's what we pick for now, but you can use a delta distribution or a power law distribution as well. And so one might ask, how can you actually do some analytical progress here? And one way to do that is to actually move to Laplace space, right? In Laplace space, you can write down a closed solution for your P. And to get the moment, there's actually sort of, by definition, we know that this ISF is the characteristic function. And so the moments we can simply get by a low wave number expansion of the propagator. So you see here, we can, once we know that, we can actually read off the moments, like the mean and the second moments and up to whatever order you like. So I hope that makes sense so far. So what we can calculate analytically now is actually, for example, what is the mean displacement? It turns out if you have all your particles starting from here and you look at cases of different resetting rates, those with a higher resetting rate show sort of like more directed motion than those with a lower one. And in fact, the mean displacement turns out to scale with lambda by 1 plus lambda, which is this reduced resetting rate. And it's along the direction of the resetting angle. Okay. So if lambda is really large, what we would get is just simply directed motion along your external field, if you like. But if lambda is small, you would have like a linear increase in lambda. So now what you already observed from the trajectories, we can look at the variance, and what you see in the trajectories is that they seem to be straighter and have less fluctuations as you increase lambda. And so this is what you can quantify in terms of the mean square displacement. So here the mean square displacement increases linearly at short times due to translational diffusion. So this is, I should say, along the x direction, along the direction of your resetting angle. Then you have a quadratic increase in time because they move actively. And at long times, it essentially gets effectively diffusive again. And a similar behavior can be observed for the y direction. So now what's perhaps important is that the effective diffusivity at long times decreases as we increase the resetting rate. Okay, so the distribution or the fluctuations become less. So now since we've seen a lot of intermediate scattering functions so far, now I just want to show you one where we actually look at a system with a drift, right? So now here in this plot, you can again see these. Okay, let me first say, now I'm going to show you the real part of the ISF, and I'll come to the complex part in a bit. So if we look at rare resetting events, okay, then you can imagine that the cells in between resetting events actually can change their swimming direction due to rotational diffusion. And so in the ISF, what you can see then the following, you see that at times that are smaller than this rotational diffusion time, we see these oscillations due to active motion. And so now these oscillations become smaller as we decrease the wave number, right? And then they start to appear again. And so when they start to become larger again, that's exactly the point where resetting becomes important. So I just want to show you that, you know, for this interplay, there's an interesting interplay between resetting and rotational diffusion that takes place. So now, if we move to a larger lambda, we know that the particles realign with the field before the rotational diffusion has sort of changed their swimming direction. And so we see essentially first oscillations, again, due to active motions, and they're immediately followed by those for resetting. And that is simply because particles cannot reorient due to rotational diffusion. Okay, so there's this idea. I mean, this might be a bit too detailed for those of you who are not familiar with this. But what, again, the message is you can detect different dynamics from these ISFs at different levels. Yes.
Sorry to interrupt. Can I ask you a question?
Sure. Please.
So, how do you measure the small lambda?
What do you mean, like
Not the capital one,
the small one, like which is the frequency to reset, or like the frequency to adapt to your chemical stimulus, for example? How you measure that? Or
say in an experimental setup.
Okay, so in an experimental setup, you could now use this theory and sort of try to fit it to your experimental measurements, and that should then give you like a frequency at which the particles realign.
Okay, yeah, thank you. Thank you.
Yeah. Okay.
Hello.
Yeah. Sure.
The question is like, then you are saying that if rare resetting is there, it is 0.5 and frequent resetting is 1.5. So how one can decide like what should be the lambda factor for our experiment?
Well, that you have to measure, right? So, I mean, in E. coli, I can tell you that the rotational diffusivity is smaller than the tumbling rate. So in that case, we would be in this frequent resetting regime. There are, however, archaea where you have exactly the opposite, where the cells only reverse their swimming direction, change their swimming direction at time scales much longer than the rotational diffusion time. But again, these are things that you can either measure by tracking probably, or by really fitting this theory to DDM experiments, for example.
Okay, so basically rotational diffusivity rate does plays role for resetting this, right?
Well, rotational diffusion plays a role in sort of all micro scale swimmers, right?
Yes, ma'am.
And so, yeah. So, and then just the way, like whatever your lambda is for your resetting, you will get different dynamics of the system, if that makes sense.
Okay, thank you.
Yeah. Okay. So perhaps one of the most important aspects here is that if you have something like this external field, you also end up having an imaginary part of your ISF. Okay, and this is what I'm showing here in these two plots. And you can also see that the oscillations are stronger if you have more frequent resetting events. Okay, so now if you end up measuring, let's say, the dynamics of your system and you compute the ISF and you get an imaginary part, that indicates that there is some directionality in the system, some drift or some motion along a certain gradient, if you like. The way that this can be mathematically understood is if you go back and think about this expansion of the ISF, you would see you get the drift exactly due to these mean displacements, right? And then you will get a complex part again due to the third moment and so on. Okay. So what we can do now is we can actually transform the ISFs back to real space, and we can compute the full distribution semi-analytically. And so here you can see a distribution as the distribution evolves over time. And I think this one here is quite interesting because you see that the distribution moves along the x direction, along the resetting direction, but you also see these features here that there's some probability of the cells that they have actually moved backwards, right, due to rotational diffusion. And if we then wait long enough, we end up with a Gaussian that like moves along the mean drift. And so we can actually also look at the comparison of these distributions. And here we just look at distributions along the x direction. And perhaps if we again focus on 0.5 and 1.5, so the purple and the green curve, you see qualitative differences, right? So for example, here in this plot, you see that if the resetting rate is smaller than rotational diffusion, you see this almost bimodal distribution, while in the other case, most of the cells are just simply moving along the resetting direction. Okay. And here I should say the symbols are from single particle simulations and the lines are from our theory. So we can really fully capture the process analytically. Okay.
Can I ask you a question?
Sure.
What happens if there is a flow?
Yeah, that's an interesting question. So a flow would tend to, I would think, well, depends on the flow. If you have a directional flow, you would keep this real and imaginary part of your ISF. You would need to think a bit carefully if you can actually compute that, analytically. The way where you would add it theoretically in your equations is in this propagator here, in this P, right? So if you know that for a situation where you have flow, you can add, if you speak to the tumbling behavior on top using the renewal equation. That makes sense?
Okay, so you're saying that this probability that will incorporate the flow.
Exactly. But this will, yeah. So this will be challenging, right? To compute analytically. But perhaps to some extent, perhaps like the moments are possible, and then you can do a wave number expansion. So yeah. Okay. So the theory is very general or generic to these aspects. Yeah. I mean, of course, if you just incorporate it here, you assume that the reorientation of the cell doesn't really depend on the fluid, which is probably not true. But yeah, one can think about extensions along that line too.
Okay. Yeah. Thank you.
Okay. Any other questions? Okay, then let's just quick summary of this. So I showed you we can use this renewal theory to actually think about a minimal model, let's say for chemotaxis. There is this interesting interplay of diffusion and drift, and we get this imaginary part of the ISF. So, and of course, these things can be pushed towards looking at first passage time properties, finite resetting durations, and also looking at this scenario in complex environments would be interesting. So this is part of Yannis Bausch's PhD work that I just showed you. So are there any questions to this topic? Okay, so now if I have time still, I can talk about a problem that relates to swimming in porous media.
Yeah, I think you have half an hour if you want to take.
So I think I'll take that half an hour. So the last part will be about, I've showed you all these examples of how microorganisms move in their habitats. And one feature was that they often move in porous media. And I'm sure Tapo showed you some of his beautiful experiments along that line. So the question that we were interested in goes back also to some really old, or like kind of old work by Berg. And what they were looking at were like cell colonies that were spreading in an agar gel. And they found something interesting. So they found that if cells don't tumble or tumble all the time, that the swarms at the end were much smaller than the swarms for cells that were able to tumble at the finite tumbling frequency. Okay, and they looked at this at the population scale, right, at the whole swarm. So now this non-monotonic behavior has been pointed out also in theory for single particle models, so that there's some non-monotonic behavior of how cells spread in porous environments with respect to their tumbling frequency. So now, in the recent past, Tapomoy and Suchit did these beautiful experiments of looking inside the porous media, and they found that these cells perform this hop and trap motility, right? And so what we were, and this is now at the single cell behavior. So what we were interested in is like, how do the dynamics of an individual, or like, how are the dynamics of an individual cell inside this porous medium related to the geometrical features of the medium? And can we, using or looking at that, predict actually what's the optimal spreading behavior? And so this is work that was done in collaboration with Howard Stone, who at the time was my postdoc advisor, with Suvendu Mandal. Did the simulations also in a group of Hartmut Löwen, and of course together with Tapomoy and Suchit, who gave a lot of inspirations with their experiments and input. So now, how do we model active agents in porous media? Well, for that we look at a simple bead spring model. So we model bacteria, essentially elongated bacteria, as a chain of beads, and on each bead there is an active force that pushes the particle forward, and the equation of motion for such a self-propelled polymer now has three terms. So first, as I said, the active force, which lets it sort of move along its orientation. There's a stochastic force because it's also subject to Brownian motion. And then we have this interaction term, and this interaction term includes the interactions between the beads of the polymer, but also the interactions with the environment, okay? So we use WCA potentials for that, and the finitely extend the finite potential also that the chain doesn't extend. Okay, so that's the first ingredient to our model, and as I said, we were interested in how the tumbling behavior affects the transport behavior. We incorporate a run reverse motion as a start. So the cells essentially move straight or move for a while. They're subject to noise, so they do change their swimming direction, and at random times they then reverse their swimming direction. So essentially the active forces just change sign, okay? And so this behavior is again exponentially distributed with an average reversal rate lambda, okay? So now we have two dimensionless numbers. One is the Pecklin number, which measures the active motion of the cells with respect to translational diffusion. And the second one measures, is the ratio to time scale. So it's the reversal rate with respect to the diffusive scale. Okay. So now in free space, the simulations can look something like that. If you have like long elongated polymers, so they move for a while, they reverse the direction and they continue moving. And so to quantify that motion, we typically look at the mean square displacement. And so here you can see the mean square displacement as a function of time, and it for different reversal rates, right? If the reversal rate is small, they almost move straight all the time. If it's large, they reverse frequently. And so here you see that at short times, the MSD grows linearly in time due to translational diffusion. We see an intermediate regime which goes quadratically in time due to the active motion, and then again at long times it becomes diffusive. due to noise, okay, and due to the tumbling events. And so what's been shown theoretically, and as I told you before, like the effective diffusivity is given, or like scales with the velocity squared by the reversal rate. So, you know, as you increase the reversal rate, the effective diffusivity goes down, right? And that's something very intuitive, because if they reverse more, they just remain within a small region, but if they reverse less, they're able to explore the environment. Okay, so now that's the free case scenario. So now how do we model the porous medium? So the porous medium is essentially just a static matrix of monodisperse overlapping spheres, and they are in size a bit larger than the polymers themselves, or than the active agents, which was inspired by the experimental setup. And so now there are a few quantities that you can look at if you want to characterize this porous medium. Well, one would be the pore size itself. That's shown here, like sort of the width, and you can measure it. But the second, and that's the more interesting part, is actually this chord length distribution. So here you can see a chord length distribution for different number of obstacles. They all decay exponentially, and they decay fast, obviously, if the environment is more dense, right? Because then there are more obstacles, which shorten the path. And these chord length distributions, or I should have said that, they can be measured essentially by putting, you know, you take 2D slices of your environment, you place straight path into those on the slices, and you just measure the distribution of those straight paths. Okay, so the relevant length scale that you can get from such a distribution is actually the average chord length, or you think about it in terms of the longest chord length, and that's something I'd like you to remember. Okay, so this is how these simulations look. Well, you don't see so much. You see that the cells sort of like hop through the environment. They become trapped like this black one for a while in pockets of the environment. Okay, so now to quantify that, we can look at the mean square displacement, just as we did before. So this are the same parameters, like the same reversal rates, just now in a porous media. And what we see is essentially that at long times, those are, again, diffusive. And we observe that those cells that never or hardly ever tumble actually show the lowest effective diffusivity, while those that do tumble show a higher effective diffusivity, and it's higher actually in terms of orders of magnitude, right? But there's one more thing that you notice. The behavior is actually non-monotonic, because the maximum effective diffusivity that you can get here is at an intermediate reversal rate. Okay, so we can measure that, and we can measure that for different obstacle densities. So again, perhaps if there are 0 obstacles, we know that as we increase the reversal rate, the effective diffusivity goes down, right? That's the black curve here. But as you increase the number of obstacles, we see this sort of non-monotonic behavior emerging. So there's sort of a maximum reversal, maximum effective diffusivity at an intermediate reversal rate. And so now the question is, so this sort of reproduces what has been seen before. The question now is like, where is this maximum? Okay, so here I'm plotting, like we use like the densest environment of those, and we plot the effective diffusivity for different Beckley numbers, so for polymers of different, that have different velocities. And I'd like you to remember like 2 important length scales in this system. The first one that I told you is the maximal pore length, right? So the maximal path that I can find in this environment. And the second length scale that's important is actually the run length of the swimmers, or the persistence length. It's the length that they move before they change their swimming direction. Okay? And that's what we call as L run. And so now it turns out, if we rescale the x-axis accordingly, the maximum effective diffusivity is exactly when is where the maximal pore length is of the order of the run length of the cells. Okay, you can imagine that in this case, the cells can actually explore their longest path without reversing and without getting stuck at the end. So this sort of led us to define this geometric criterion for optimal spreading. That cells are generally expected to spread most if their run length is of the order of the maximal pore length. Okay, and now you want to probably ask, what are the dynamics of the cells when they move in these environments? And so what we can look at are actually the trajectories, right? So we have here the position, so one coordinate, like the x position as a function of time, and they show that for, yes?
We lost you for like last 10 seconds. So,
oh no.
Yeah. Sorry.
Okay. So
after you started talking about trajectories.
Okay, so I'll start with the trajectories then. Yeah. So we can measure these trajectories, right, for different reversal rates. And here I'm showing you the plot where you show the trajectories of one coordinate as a function of time. And we see quite different behaviors as we change the reversal rate. So if we have a really small reversal rate, you see that the cells seem trapped for very long times, right? And they can only hop very, very rarely. So now if you take lambda really large to 15, for example, we see a lot of like forward and backward motion, if you like. So they're really small trapping phases, but the displacements are also not very large, right? And at an intermediate rate, now we find that, well, the trapping phases are shorter. Also, the hop length are much longer. Okay, and so you can see from these trajectories, you would already expect that this intermediate one sort of spreads furthest in the environment, right? So there's this trade-off between trapping and hopping through the pore space. And that's something that has been already really found in experiments. And we now just try to quantify these dynamics in terms of a coarse-grained model. But before we can do that, we can actually look at the distributions of these hopping and trapping phases, okay? So let me first walk you through the trapping time distribution. So here we really measure the trapping time of the cells. And you can see that the distribution are sort of power law distributions. And one can really measure that the average trapping time increases. So these are the dots here as you move towards really small reversal rates, right? So the cells are trapped for much longer time if they cannot reverse. And this is a change by orders in magnitude, actually. And then what we can also, and the exponents here of the trapping time distributions are sort of in agreement with those of the experiment. And the second quantity, we can actually look at it at the distribution of hop length. So how far do the cells hop before they either get trapped or before they reverse, okay? And also here we see this like non-monotonic behavior of the average hop length, which peaks at an intermediate reversal rate, okay? So and the way to understand this is like if the reversal rate is really small, the polymers just move forward and backward in the same pore. If it's large, they move forward and backward in the same pore. But if it's really small, the hop length is really, they cannot essentially explore the environment because they just remain trapped. Okay. So what else did I want to say? Yeah, I think that's about all. What's important to note perhaps is that you can see that the distribution of an intermediate reversal rate shows this like sort of rare events, or like there shows sort of a larger probability for longer hops than the others do, right? So these sort of events where the cells can hop for a hop across a hop further, let's say, actually contribute to this larger effective diffusivity. Okay, so finally, let's just try to model this in terms of a coarse-grained model for these hop and trap phases. And the way we thought about it is very similar to what I showed you for run and tumble particles. So we wrote down a model that is similar, or like in terms of these renewal equations, but now that the processes are a bit different. So during a hop, we assume that the cells hop for an average time at a certain velocity, and they reverse during that time, okay? And then once they become trapped, they become trapped also for an average time. And these two processes, of course, also depend on the distributions that I've just shown you before. So for the trapping events, we use a power law distribution, while for the hopping time, turns out they're exponential. So we have an exponential distribution. So now we can sort of like look at that model, we can compute the moments, and from those we can extract actually a long-time effective diffusivity. And that turns out to be of this form. So it depends on the fraction of time that cells spend hopping. So tau h is the time cells spend hopping. And then it inversely depends on the reversal rate, right? So now you can imagine if the cells are trapped for a really long time, the effective diffusivity becomes really small. And at the same time, if they reverse very frequently, the effective diffusivity goes down as well. So now lambda is an input to our simulations, and we can measure tau h and tau t from our simulations, from the distributions that I showed you. And so now with these parameters, we can actually sort of predict the behavior of the effective diffusivities up to a prefactor, actually. Okay, so now that means that, if you have frequent reversals, the diffusivity goes down, but also if you have long trapping phases, so this whole non-monotonic behavior is an interplay of these two mechanisms. And with this, I just want to point out that one can think about the generality of this criterion that we just showed, and one aspect to modify the model was actually to look at concave pores. So now in this case, the cells, instead of moving in the void between the spheres, they move inside the spheres, if you like. And also for this type of model, we sort of really find that the geometric criterion still holds, okay? And then what you can also do, you can think of, well, now you did run reverse, what about run and tumble, actually? Run and tumble in terms of like, what if the cells can actually change their swimming directions randomly in 3 dimensions? So we sort of extended our model to that, where we essentially at random times kick the polymer ends along different directions, and that sort of mimics a run and tumble behavior. And also in this case, we find that the geometric criterion still holds, and we have an optimal spreading behavior when their run length equals the chord length of the environment. So with this, I'm at the end. So I showed you that we see this non-monotonic spreading as a function of the reversal rate. We can think of it in terms of these hop and trap dynamics. And we identified this geometric criterion, which sort of remains irrespective of details of the pore shape or reorientation mechanisms. And with this, I'm happy to take any more questions if you have. And thank you all. Well, perhaps before I go to the acknowledgements, let me just make some advertisements. Well, first of all, we just recently published a book on these topics, on out-of-equilibrium soft matter, active fluids, which contains a lot of interesting chapters from very interesting authors. And then second, we have internships, an internship program at IISc for mostly undergraduate students. So in case you have any or know about any people who might be interested, please forward this information. And what's perhaps more relevant for you is that we also have several calls a year for postdocs and guest scientists at IISc, and the deadline for the current one is actually next week. So with this now, I'm at the end. I'd like to thank all my collaborators and mentors and my group and all of you for your attention. So.
Christina, thank you, Christina.
Yeah.
Is there any question? So,
hi.
Hi.
So instead of this porous medium, if you have a glassy medium where the cage also relaxes in finite time, do you?
I didn't.
Sorry,
hear
the microphone stopped working.
Okay.
Yeah. Do you expect the power law distribution of trapping time to hold? And do you expect that there will be a non-monotonic spreading?
Okay, this is a very interesting question. There is one paper that deals with active glasses. And in that case, they find that also this persistence length is the defining length scale in the system. This is Lisbeth Janssen from the Netherlands. I don't think, well, regarding the trapping times, I'm actually not sure. One would need to look at it. It depends probably, well, for sure at the, there is another time scale, right? That sets the motion of the beats then, and perhaps.
Right. So you might also expect an exponential tail.
Yeah, it could be a combination
Yeah,
of.
could be a combination. Okay.
Yeah, exactly.
And okay.
Yeah.
Any other questions? Okay. Yeah, one more. Yeah.
Hello. Yeah, have you looked at apart from bacteria, any other cell in 3D, say some mammalian cell? I mean, maybe the motion is different, but.
You mean mammalian cell in a 3D porous medium?
Yeah.
No, we have not looked at that. I think in 2D, actually also Tapa and colleagues, they looked at at polymer rings as a model for, I would think, deformable cells in 2D, active sort of rings.
Yeah.
So you can have a look at, maybe Tapa can comment on that. In 3D, we haven't done it, and I'm not aware of any work that did it theoretically. Yeah, but one would need to account for probably for the fact that the particles, that they can deform. That's probably a large issue. Some of them might perhaps only move because of the boundaries, so it's probably a very different setup. But maybe Tapa can say something.
Okay, yeah.
Okay.
Yeah, so I think we looked into in 2D, but in 3D you have also volume fluctuation and stuff like that comes in, but not also us, but also Alice and Patterson. They did this bilayer models of active ring systems where they look in model the vimentin layer and when cells squeeze through different constrictions and stuff like that.
Okay.
Can you go to slide 10 where you show differential microscopy?
Yeah, sure. Right. Okay, I'll go. Yes.
Here, for what, what is your, for what are you taking this microscopy here? I think I missed that. Like the images that are shown.
Oh, these are
the
simple bright field images or phase contrast. There's nothing fancy about the images, if that's what you are asking. You can take any. You can take also images with fluorescently labeled particles.
So I think the question was of what? I think this can be any particle.
Oh, that was the question. Sorry. Yeah, yes and no. So of course, like if you have, well, we assume E. coli to be sufficiently point particles, but you can also have effects from the particle shape. Yeah.
So you think like if a particle changes shape, like as you would have picked up in the other scattering forms, you will also pick if I have static systems but changing shape, it will pick up that through this formulation.
Yeah. So what's encoded in this function are intensity fluctuations of your sample.
Right.
So if you have a shape change, you will have that also coming up in your resulting function, I would think. But how to analyze that, I'm not sure in terms of theory also and what it tells you. Yeah. I mean, people have looked at, for example, just in terms of shape, they have looked at, for example, filament suspensions. You have a network of filaments and you have beads in it, and then you can look at both the motion of the filaments and the beads at the same time if you label them differently. And you can get some relaxation times out of these signals at the end. Or even for the particle, you can get some exponents, I think, for the MSD.
Oh, that's amazing. Okay, I'll ask you later about that.
Here, if I look at the IRT images, these look similar. But if I look at these GQT, those images, like which shows the correlations, why the intensity is increasing as like for T1 to T2 and further.
Okay, these are just simple gauge, like so the images I'm showing here are not having any actual meaning. So it's not related. So this would correspond probably to exponentially relaxing things, to diffusing particles, I think. I'm not sure. Yeah, I'm sorry. These are just like for purpose of
yeah, so it just says like if you look into the inverse space, you get a scattering function. That's all.
Yeah.
Any other questions? Well, then let's thank
Madam,
Christina. Oh, one more. Okay,
sorry.
Go ahead.
So, madam, how would you model such systems, or how would you choose the waiting time distribution where system is in ballistic regime at very low temperature, which we could potentially see in every system? Then at intermediate regime, it shows some kind of anomalous diffusion, and as usual, it shows normal diffusion at long time. So how do you model such system, or you can how do you choose the waiting time distribution there?
I mean, you could
actually excuse me. So actually I am talking in continuous time random perspective. So for this waiting time distribution to be exponentially distributed or Poissonian distribution, then the power, I mean this MSD shows a normal diffusive behavior, and whereas it has a power law distribution, then we could have subdiffusion or superdiffusion. That is the anomalous diffusion. So when this transcendence happens from anomalous at intermediate regime to normal diffusion at high regime, I mean high regime means at long time, then how would you choose this waiting time distribution in such?
But that's an interesting question. Well, you can look at I haven't analyzed this, but if you have, like, let's say a power law distribution of your waiting times here, I could imagine that you get some intermediate regimes that are not diffusive, that are somewhere in between ballistic and diffusive. Yeah, I think you have to do some. There are some theories, obviously, where they look at these power law distributions in these contexts and they find these different behaviors. Yeah. I
So
don't
we
know.
can't choose a single time distribution to show a whole pattern, right?
Yeah.
Okay, thank you, ma'am.
Okay, in interest of time, let's thank Christina for waking up so early and giving this talk even beyond time. So
Thanks for having me, and I hope you have a great school.